Most QSP projects don't stall on the mass. They stall on the data, weeks of it, before a single equation gets written. I'm Vladislav Leonov, database manager at InSysBio, and this is how we take the months of your timeline. Two databases, CYTOCON database for in vivo concentrations, fIVE for in vitro kinetic parameters, and an annotation service for everything they don't cover. Quick map of what we'll cover. First, the problem, why manual literature curation eats so much QSP project time. Then how we built the quality control pipeline behind both databases. Then deep dives into each database with a live demo. We close with the annotation service, what we do when your specific question isn't covered by the standing databases. If this slide doesn't describe your situation, the rest of this talk probably isn't useful to you. Every QSP project starts the same way. The modeler collects data, PubMed, reading, pulling values from figures and tables, sometimes literally clicking on PNG images, converting units across studies that each used slightly different conventions, trying to figure out whether two values from two different papers are even comparable. On the left, 5 things QSP modelers tell us repeatedly. Weeks on PubMed. No unit standardization, no parameter formatting for ODE models, gaps in coverage of specific diseases or tissues, and a real error rate when you're extracting manually. On the right is what that actually costs, based on what teams we work with report, 4 to 8 weeks for initial data collection on a typical project. 30 to 50% of early project effort spent on data instead of modeling. And it's recurring. Every new project starts essentially from scratch. The databases we built are a direct response to this. Before I show you what's in the databases, let me show you how data gets in, because if you don't trust the pipeline, the numbers don't matter. 4 steps. Applied to every entry in both CYTOCON and fIVE. Markup. A PhD level reviewer reads the source article and decides what should be extracted. Values, metadata, units, patient characteristics. This is a human with domain expertise making editorial decisions, not a script. Annotation. A team member, all with degrees in biology or medicine, transfers the marked up data into the database. Raw values stay raw. We record exactly what the paper reported plus context. Review. A second annotator independently checks for accuracy, completeness, consistency. If something doesn't hold up, it goes back to markup. This is where extraction errors get caught. Error detection. Algorithmic. We scan every new entry against everything we already have for the same species, tissue, and disease. Outliers get flagged. If it's real biological variability, the range adjusts. If it's a misprint or extraction error, it's removed. Same gate for both databases. This is why we can give you values you can actually put in a model. Let me make this concrete, and I'll carry it through the whole demo. A modeler on our team is building a severe asthma model. The goal, capture the eosinophil axis mechanistically, validate it against drugs that already target eosinophils, then predict what a new compound does. To build that one node, she needs 2 things: how many eosinophils sit in asthma patients and what they secrete and what controls it. In vivo numbers, in vitro kinetics. Normally, it takes weeks in PubMed. Cell and cytokine concentrations database is our in vivo concentration database. It answers the question, what's the baseline level of cytokine X in tissue Y in patients with condition Z? We covered around 500 disease types with more than 100,000 individual values, extracted from almost 3,000 papers. Everything in unified units, kilocells per liter for cells, picomolar for molecules. You can compare across studies without doing conversions yourself. We update monthly, around 1500 new values. In QSP, you fit as few parameters as you can. You ground what's measurable and reserve fitting for what's unidentifiable. Every baseline you pull from literature is one less free parameter, less overfitting, a model you can defend and review. CYTOCON hands you those, with the unit consistent and the source traceable. This is the CYTOCON DB landing page, featuring a description and options to sign in or try it free. The free version, open to everyone, offers the same core functionality as our main product, but with limited content. Contact information is also available if you have questions. Clicking Supplementary Materials gives you details on unit conversion, molecular weights, and tissue parameters. There you'll find a full description of how original units are converted to our unified standard, including the specific formulas and reference sources used. Special units cover cases where data is given as a percentage of a parent population, like CD4+ cells as percent of CD3+. Molecular weights for molecules conversion. Tissue parameters like interstitial fluid volume and skin thickness used for conversion. And of course, you'll find details on our algorithm for weighted mean and standard deviation recalculation. In additional info, you'll find detailed content metrics, the full list of disease types, tissues, cell types, and markers currently in the database. This gives you a clear picture of coverage before you start querying. Click on any disease to see its specific parameters, including which tissues and cell populations are represented, and a full description of the condition itself. We currently host a substantial number of diseases, spanning multiple therapeutic areas, with new ones added regularly as the database grows. You also can find our API section with complete documentation for remote database access, letting you integrate CYTOCON directly into your own pipeline or software without needing to use the interface manually. You'll also find general information about us and a description of our update cycle. We release new content roughly every month. Now let's log in. Once inside, you'll see a brief explanation of how to work with the database. Clicking here opens the full manual, which walks through every step in detail. I'll also go through these steps manually today so you can see exactly how it works in practice. It is time to move on to the query section. This is our query interface. You can start from pre-built requisitions. Today, though, we'll build a specific query. We will look for cells. Disease field set to asthma. Species set to eosinophils and activated eosinophils. And tissue compartment will be blood. Note the calculated weighted statistics option. Use it cautiously. Without specifying a single tissue, pooling data across multiple tissues will significantly skew the weighted result. After clicking Query, we will get about 134 cell concentrations for the asthma patients in blood. Enabling weighted statistics collapses this into one line. Species, arithmetic mean, standard deviation, the marker list, applied unified units, tissue type, and the diseases included, since asthma queries also pull related terms like allergic and bronchial asthma. Each entry also links to its PubMed source and our conversion methodology. Now let's remove the tissue filter to see what's available across all compartments. For cells, you can activate cell markers using AND or logic. There's also a disease severity attribute covering phase, severity, or status. Additional attributes include patient number and age. The query interface also might include other filters to help stratify patients more precisely, and these vary depending on the specific disease and species entity. Once you're satisfied with the query, results can be downloaded as a spreadsheet for further analysis. Here's the cleaned up summary table for eosinophils and asthma. We're covering 8 asthma subphenotypes, with all values standardized to kilocells per liter, 310 total values, drawn from 90 unique papers across 10 tissue compartments. Blood accounts for the majority of values, followed by induced sputum, and bronchoalveolar lavage. The remaining compartments are represented more sparingly. These arithmetic means are unweighted and pooled across heterogeneous sample matrices, useful for assessing coverage but not directly cross-comparable, since compartment-level harmonization happens at the annotation service step. For actual analysis, you'd stratify this data by severity and other patient parameters, depending on what the specific study requires. In vitro experiments database is built around quantitative cell behavior. What concentration of drug X causes cell death? What's the proliferation rate under cytokine stimulation? What's the IC50 in a specific cell line? We have parameters for migration, cell death, proliferation, differentiation, with majority for secretion calculated from more than 800 publications. This is where you go when you're parameterizing an ODE model. IC50s, EC50s, kinetic constants, values from controlled in vitro experiments on model cell lines or samples from the patients. We delivering around 800 new parameters per month. This is the landing page for fIVE, following the same logic as CYTOCON. Sign in or try the open version for free. Here you'll find a full breakdown of database content, comparing what's included in the full version versus the open version, so you know exactly what you're getting with each. Our contact details are also available here if you have any questions. Let's sign in. You'll see a similar short instruction. Clicking Query will take us to a new query interface. Under Parameters, you'll find base constants, IC50, EC50, Emax values, and other, along with a list of regulators and process modifiers present in the database. Cellular setting includes cell type, cell line, and source compartment of the cells. Process settings include process type, such as proliferation or excretion, along with cell product selection and daughter cell differentiation. Finally, patient description, including patient state and age. So let's build our query. Patient state set to asthma, cell type set to eosinophils. Then you can further specify parameters like compartment and specific process. But let's first see what we have in general. As you can see, we have 23 items available for this condition. Each entry includes the producing cell type, the process involved, units, and parameters. You can download all of this as a CSV file containing the full relevant information. Here's the cleaned up table: parameters, values, and what's actually being secreted. In this case, eosinophils and asthma secrete three products: IL-13, TGF-beta1, and IL-11, drawn from two source papers, giving us 23 records total for one cell type. For each product, we have baseline secretion rate, Kbase, along with Emax and EC50 values describing how different regulators influence secretion. For example, GM-CSF, IL-5, eosinophil chemotactic protein, and complement component C5a all modulate IL-13 secretion by eosinophils, each with its own Emax and EC50. Similarly, IL-17A, IL-17F, and IL-23 regulate TGF-beta1 and IL-11 secretion. These values can be used directly from mechanistic modeling of the secretion process itself. But now we're interested in a related question. What are the actual concentrations of these secreted products inside the human body? So let's go back to CYTOCON. Now we set species type to molecule and disease type to asthma. We are trying to set species to IL-13, TGF-beta, and IL-11. Unfortunately, no data is available for the last one. In the end, we have 64 items. Let's download the CSV. So here's the outcome of the three eosinophil secretion products DAC identified. IL-13 is well covered in vivo and ready for use. TGF-beta1 has just a single record, and IL-11 has no in vivo data at all in any compartment. This kind of gap is common, and it can often be mitigated. For instance, by searching healthy controls or closely related diseases where the same molecule may have been measured even if asthma-specific data is sparse. But this also raises a broader point. A database alone isn't always enough. Both databases cover a lot, but neither covers everything, and that's the honest answer. 3 things you'll hit. 1. Coverage gaps. No database covers every disease, every tissue, every paper published last week. If your project is on a rare condition, a novel modality, or very recent biology, your topic might simply not be in scope yet. Finding what's missing puts you back on PubMed yourself. 2. Useful data outside our scope. Our databases focus on concentrations and kinetic parameters, but your model may also need expression levels, transcript abundance, or related markers. That information often sits in the same papers, we just don't curate it. 3. Pooled data doesn't always match your cohort. You might need values stratified by sex, age band, disease stage, or experimental approach, not a population average. And your equations may use units we don't ship in. Each of these costs weeks of manual work, unless someone does it for you. The annotation service is the same pipeline I showed you on slide 4, same PhD reviewers, same quality gate, pointed at your specific question instead of at the general database scope. We rerun the search and fill the gaps. We curate the literature specifically for your topic, including what isn't yet in CYTOCON or fIVE. You don't repeat the search yourself. We flag adjacent data. While we're going through the papers, we'll surface expression levels, transcript abundance, related markers, anything potentially useful. We don't process them. We point you to the source so you can follow up. We stratify and convert to your spec. Cohorts split exactly as your model needs: sex, age, disease stage, experimental method, values delivered in your units, not just picomolar or kilocells per liter. If your ODEs use nanomolar, we ship nanomolar. Turnaround: about 30 days from project start to delivered dataset. Let me walk you through how our annotation service runs start to finish. It begins with your request. Whether you specify the topic, required tissue, cells and parameters, or the specific cohort's description, we start by identifying the gaps in standard databases and sourcing the latest data relevant to your question. Here is the core of our approach. Instead of a 30-day black box, we provide a mid-project report. This allows you to review our progress, provide feedback, and if necessary, adjust search criteria or pivot the angle entirely before we finalize the work. In the final 3 weeks, we complete the annotation, run it through our quality gate, and prepare the delivery. End to end, the process takes about 30 days, with every value quality controlled by the InSysBio annotation team to the same standard as the databases themselves. One more thing on the annotation service: it doesn't have to be applied only to public literature. Many of our pharma clients have internal data they can't or won't share publicly: clinical trial values, proprietary measurements, internal repository entries. We can run the same curation pipeline on that data under NDA without anything entering CYTOCON or fIVE. Your data, never our database. Same curators, same harmonization, same stratification applied to your internal files. Nothing crosses into the public databases. Contractually and technically protected. ISO 27001 Certified Information Security, the recognized international standard for information security management. Access limited to the assigned team. And we already do this. Multiple pharmaceutical companies, big and small, currently work with us under NDA on confidential datasets. This is established work for us, not a new offering. Public literature or your proprietary files, same framework, same quality, your data stays yours. To wrap up, here is how to choose between the 3. CYTOCON database. Use this for in vivo baseline concentrations that can be used for virtual patient calibration and defining baseline ranges. fIVE database. Best for in vitro kinetic parameters for ODE parameter identification. Annotation service. Choose this when you need a custom data set or need to fill specific gaps not covered by standard databases and have data in more convenient format. Our databases are available by subscription with a free open version, while the annotation service is delivered on a per-project basis with a 30-day turnaround. That is all. Thank you for your attention. If you have any questions, please get in touch with us.